IPM for apple scab. Let's check it out. Okay, one of the first things you can do for all of the apple diseases that I'll talk about is always plant using the best horticultural practices. This is the most important thing. This put your best foot forward. And in this regard, and I've talked before about the high density plantings being maybe more susceptible or more easily devastated by fire blight, but overall, these are the best things to do for disease in general, including fire blight, because of the other reasons. They're better for color, they're better for yield, they're better to get your applications on. I find myself that these things are a dream to spray compared to the tall spindle semi-dwarf planting that you see on the left. Both of these orchards are actually in China, but you can imagine looking at which ones are going to be better for coverage, which ones are going to be better for drying time, which diseases hate, and which ones are better for air circulation. It is always that tall spindle planting over the semi-dwarf, over the vertical axe that's a little more bushy. You're just not going to be able to get the same level of control. In many instances, your best fungicide that you can never get resistance to is air. And always grow the best possible orchard that you can. And then the disease control will come naturally after it. And that along those lines, water management's important. Pick the best sites. If you're getting to the bottom of an orchard area, the bottom of a hill that might flood, just don't do it. Make sure to tile orchards. Make sure to do a good job of your drip. If something bad happens and the drip line explodes or you have a wet area and it just gets weedy, all of this is just going to put you off on the wrong foot for disease in many instances. Weeds are important. Water management is incredibly important. All of this will make drying more important. And drying is one of the best things you can do to stop all diseases, whether it's fire blight, whether it's apple scab, that we'll talk about next. And then here's our apple scab. It's a perennial problem in cool, wet production regions. If we begin to migrate away from cool, wet portions of the season, we'll be able to have an excellent handle on apple scab, and you'll be able to control it with biologicals. But unfortunately, this is what it looks like. This is some of the really intense, heavy, thick, scabby lesions on the leaves, and this is where all of your inoculum will come from in the spring, and these are the thick, velvety, scabby lesions on the fruit. This will have your entire fresh market harvest chucked in the bin. You may be able to tolerate a little bit in terms of cider apples, but for the most part, this isn't what you want. It can make the apples deformed. They may not taste or develop correctly, and they may not even be that good for your cider as well. Unfortunately, a lot of the cultivars we like to eat can get apple scab. The ones that are resistant to apple scab aren't very exciting to grow or appealing to the consumers. And in many instances, this is a high input system that can take as many as 10 plus fungicide applications per year, depending on how much wet weather we have. And unfortunately, we have a lot of fungicide resistance and nearly all the really fancy single site fungicides that can go right in there, hit genes inside fungi, shut them down, and cause them not to grow, while at the same time not harming animals, birds, and other things in the environment. So we're losing these, but we still have a few. So how does apple scab survive? First, it likes to overwinter in infected leaf litter. All the leaves have fallen to the ground, and while it looks barren and there's nothing that could possibly live here, there is apple scab overwintering and surviving the winter hidden in those thick leaves on the orchard floor. And this is, if you were to take a cross section of a leaf, this is what you would see if you'd buy one of these fancy prepared slides that I have and take some pictures of it under the scope. This is the leaf lesion right here. This is the structure, and inside this structure are the infamous ascospores that you constantly hear about. In the spring, these will mature and get blasted into the air. But interestingly enough, they don't have the forceful ejection mechanism. They're a thicker two-celled spore, and they're not going to get picked up in clouds and carried everywhere. What they're going to do is fire up directly underneath your tree, back up into your tree. And yeah, they can blow around a little bit, but they're not like some of these rust diseases. And so that's how the infection gets started. Later, when they do cause an infection on your fruit clusters, they'll make a second type of spore, sort of a lumpy spore that looks like a spore from the fungus that you might find in your bathtub. Very closely related to another species that's in bathtub molds, and this will be produced in the second lesions, and this can also spread around and cause infections as well. And for the most part, spread is very local, and management is site specific. So anything you find with resistance to your site might not apply to your neighbors. And so everything you do in your orchard will determine what levels of resistance and what types of scab strains can get loose and cause trouble. And so it's always real important to know that what happens in your spot isn't the same as everyone else. So take a look at this next scene. We'll show you some different tools and how they might work to manage apple scab in a theoretical sense. If we look on the right side, we can see some of the cartoon of the same sorts of things. You've got the leaves on the floor in the winter and in the early spring, and depending on when you get into the orchard, this is when you might make your urea application to tear up that inoculum. And at this time, the pseudothecia is developing and maturing. And when its ascospores are flying into the air, it's right around the same time as a green tip. The pathogen is sort of evolved to sort of mature at the same time as your apple buds. In particular, it really likes to mature along the lines of the cultivar McIntosh. So anything closely related to McIntosh, we've planted so much of it in New York over the years that the apple scab evolution is sort of matched up to McIntosh. If you have things that are earlier or later, you can escape a little bit of that. But one of the best things to do is to put on this silvertip application of copper. Sometimes these later spores called Canidia can overwinter inside buds, and when you put that copper on, you get some early apple scab infections, and all is well. And so this is what would occur next. You get some lesions on leaves, and then develops the fruit lesions, and these constantly infect, and this is what the fruit lesion or leaf lesion might look like in the secondary spores. And then all of this happens over the entire summer until it gets too warm, and then the leaves fall on the ground and start the process over. But if you wanted to see how your practices might actually impact the disease, anything that you do early on will impact the initial inoculum. And sort of from an epidemiological standpoint, you might have apple scab on the left axis, and you might have your time to harvest here. And so these things that you do, such as urea, silvertip copper, or the both of them, will take what this is called a disease progress curve. And this is the part here where you get to that pedifol area where the disease can have these many secondary cycles and just ramp up like crazy. This grabs this curve and pushes this down. And you are just taking the inoculum and just getting rid of it before it can cause disease. And so it really starts your disease much later in the season, to a point where once apple scab does get going, now you're practically at harvest. And in a real world sense, you would never have 33% incidence. And if you were to do it right, you could almost, in theory, if it was a dry season, delay this epidemic so much that you wouldn't need to even do any later applications at all. And that's why we've always been pushing over the years to really go after that early season inoculum. In many years, I've done trials where I've just done spring fall urea and copper, and unfortunately I'm angry because it was so dry that the check trees never got any apple scab, and that could be the case for you as well. So you can really use that to your advantage. And then, okay, after that part, what is the other fungicides doing? Once I may get to the end of this primary infection and all the ascospores have been blasted into outer space, as the models have said, once you start going to green tip to bloom, you're protecting against the new infections, which would, sort of slide that curve a little bit. But once the infections have occurred, let's say around pedifol and that first to second to third cover, when we might get extended rains, you're protecting against any secondary cycles. And what happens in this case, you may hear about these things like we got to flatten the curve, as with popular virus pandemics that may be going on at any given time. And that's what you're doing. You're taking these secondary cycles. And when you put on some of those protectants, if something slipped through and they try to germinate as these Canidia, you're taking that curve and not making it exponential. You're starting to flatten it. And over time, as you continue to put on these fancy single site, you're taking that curve and squishing it. And then you keep going with anti-sporulants that keep these lesions from sporulating. You're just grabbing that curve and flattening it to the point where if you do both of those two things, you don't have 60% apple scab at harvest. You have 0% apple scab at harvest. And that works very, very well. And so that's what you're doing. You're taking that exponential growth phase and just sort of making it flat. You're excluding apple scab from your orchard in the first part, and then whatever's left over, you are smushing it and keeping it down so that it can't grow and reproduce at a rapid rate. What else can you do? you can try some resistant cultivars, and these will work really well. And here's a bunch of different ones that are fairly popular: Gold Rush, Williams Pride, Liberty. And if you're going into organic, these are great, but they don't get all the other diseases. They really only get one, and you'll still need to apply some applications. And one of the things that's really cool is that Honeycrisp has a lot of resistance to apple scab, but this doesn't let you escape so much from the from all the other different problems and challenges that Honeycrisp has. And that's right. Many of these give immunity to apple scab, but they don't give the other diseases. For example, the cultivar Topaz, the scion is so susceptible to Phytophthora root rot that it may skip over your resistant rootstock and completely rot the tree right above the graft union. So good for apple scab, not good for everything else. And as we've talked before, sanitation is important, and this is one of the best things to do. Get in there, get rid of those fruit drops, get rid of the leaf litter, any dead prunings. And, a lot of this I mentioned before, apple scab is on the leaves, but all of these other pathogens hide on fruit drops and leaf litter and prunings. And anything you can do avoids that buildup of inoculum. It's taking your disease progress curve and just pushing it off the end until you get out of harvest. Here's a nice picture. Here they have a machine in Brazil that sweeps all the leaf litter and fruit drops in the middle, and then they have another one that comes through and mows it. You can see what it looks like before and after, nice and bare. And what you can do, fall or spring, depending on when you can get into the orchard. If you can't get in the spring, do it in the fall. You can sort of rake everything in the middle, scalp the sod, put down your urea application, which will take all those apple scab lesions and just dissolve them into nothing. Or you can do dolomitic lime. And then on top of that, as you get back into the spring, once those buds break and open up and maybe loaded with conidia or apple scab ascospores are thinking about shooting off, that's when you hit it with the delayed dormant copper. You may not want to use a monstrous handgun like we're using in this picture, but you really want one of those heavy, this is your chance to use a high percentage metallic copper equivalent. That's what that means. And something like one of the ones that are generally powdered form are often stickier, will stay on your trees a little bit longer, preventing you from having to keep going into the orchard, and often have 30% or 15 to 30% copper in them whatsoever. And so that's how that works. And we're back over here again. And then once we begin, we've sort of talked earlier about the various overwintering and the primary infection phase. And then here, once you're getting into this green tip to bloom, this is when you go in every 5 to 7 days, depending on what the weather looks like. And you may use some Captan, Mancozeb. This is a good time for the protectants. Your sulfur if you're organic, or, we can still even go back and use Dodine, which is an incredibly potent material, but you only get it twice until you get to pink. And in that case, there's a variety of different Dodine products out there, but that is a good AI to use early on if you have a big, heavy rain. And then at that point, this is the part where you're going to take the infection curves and flatten them. You're going to use this part to push it out until harvest, and you're going to use this part to make sure that your curve is very flat and there's no exponential disease development. And this is where your fancy single-site fungicides come in. From that bloom to third cover, this is when the possibility of these infections circling and constantly infecting, making new infection comes up. And this is where your DMIs, your QOIs, and your SDHIs come in. So you can forecast. And if we keep having more erratic weather where we have periods of heavy rain and dry weather, forecasting may be more important. And you may be even able to do things that are going to sound as crazy as skip sprays in the future. Could have done it in many of our trials and still gotten away with a complete success. And we have better precision agriculture and ability to be more agile and with potentially drier periods coming here and there, and we can better see the massive rainstorms coming. We might be able to get away with this. And particularly in the early season, we have good models that predict the ascospore maturity, the release, and the conditions for that first infection period, that green tip to petal fall. And we can kind of track how much has happened. Now, what's really interesting is that repeating phase, or all these conidia here that are produced that are constantly infected, is technically not predicted according to the scientific literature. But what we do know about it is it's also a spore. It's also an apple scab spore, and it also likes wet weather that's also cool. Interestingly enough, if it gets too hot, it burns those out. And even though we haven't done a lot of studies with conidia infection, anytime you see a prediction for a lot of leaf wetness and a lot of cool weather conditions, you can pretty much assume that the Canidia are going to be very happy and are also causing an infection. And I often myself use those models way out into the later parts of the season to see when I might avoid things like pinpoint scab, which can come in right before harvest. And what we have for growers is the NUA system. It's free. You can find it here. It's for forecasting apple scab. We've got a couple of different features. We have, it tells you how much is predicted as mature. And what I would always think about this as having a good degree of variability. The discharge events, I think, are pretty good. But the maturity, we know, has a lot of error associated with it. You might think, the model says I have 18% mature and I already have scab on the trees. And that's why we take a little bit of care in going out with some of those preventative practices like the urea and the early season copper, because there's a lot of noise in it. And while these things work really well in growth chambers, you're never prepared for the environment. But they work pretty well and they will give you a good idea. It's just that you wouldn't want to consider them as literal fact, but strong suggestion, if you will, of when the problems are. And so they'll project the maturity, the amount of discharge, and the cumulative discharge. And in this particular instance here, it's too late for you, but it will also show it in the future, which is pretty cool. And it will provide you warnings. And what we've done with the NUA system is the meteorologists have provided more weight to water predictions 2 to 3 days closer to the current one and less weight on water predictions further out. As we've all known that every time it tries to predict rain, it's usually most accurate 1 to 2 days out instead of 4 to 5. But sometimes you'll know when it says the big storm is coming, and you can prepare yourself for these types of things. It uses both local data. You can get your own weather station plugged in, and it will use satellite weather data that's gridded to your region. And it will use a leaf wetness algorithm. The leaf wetness algorithms will predict a lot more heavily than others. And here's a little close-up of the different shots here, and you can kind of see what was going on. In this instance, if we look at the current day, it will sort of show you, oh, goodness, a big infection. But afterwards, if you had a spray on and you were ready to manage this, you know that it was probably best to have put it on 5, 8, or 9, and then you don't really have to worry about apple scab for the rest of the week until the next week. You can see that in the future over here, really not much going on. You're in the green. There's no discharge predicted whatsoever. And then there's your big event. And as we move on ahead, here's an example of how it might be used in forecast. You might think, okay, great. I'm at the end. I'm 95% matured. Discharge has been up to 27%. Uh-oh. That means there's about 70% left to actually go. Everything's mature, but we haven't had an infection that day. And it's been 3 points, but then look ahead, it's predicting almost 30% of my ascospores. You might think you're finished with scab, but what's actually going to happen in this big storm that's predicted further out, this is where 30% is going to go, and it's going to jump up from about 30% of everything having already ejected to everything nearly being ejected. So this box tells you how much is out there that's ready to go. This box tells you how much is daily going to be ejected, and the final tells you how much cumulative over the entire season has already gone. And so you can use this to really predict the big ones. And this is where you want to go in with the special fungicide you've been saving for that rainy day, because the rainy day is coming. What else do we know about it? The predictions are really about the primary phase, the things we refer to as the ascospores, the infection and the dynamics, and it gives you some cursory information. But as I told you before, there's a lot of noise in these systems. And in many instances, if there's a lot of noise, it's not going to really predict the fine points of the model, like, is my ascospore actually germinating? Is my canidium germinating? What is actually happening at a microscopic level? There's a lot of noise in predicting these few simple things, maturity, discharge, and cumulative discharge. There's going to be a lot more noise when you try to ask more detailed questions about what's happening about the biology. And it uses forecasting data more conservatively. Less weight on 3 to 5 days out than there are on 1 to 2. And you know from the Weather Channel and all the other types of data sources that you get that that's often is not a bad thing to have. What you don't want to do is to be fear-mongered into spraying 7 to 5 days out every time and then realize the storms go away, which can be very frustrating and costly. They just really predict favorable conditions. You apply at that high-risk period. Don't go for every infection. look for the infections that are 34, 35, and that's when you do the big one. If you want to try a biological, you wait until it says, well, maybe not 9%. 9% is still pretty intense. But if you had a good fungicide here, you might think, well, maybe I'll go out and use something that's a little lighter, less hard on the environment, and make sure I get that last 10. And that's how you can sort of use these. Don't go every time it screams infection period. You'll end up wasting all your money, and you'll control apple scab, but you really won't need to make that many. And always use common sense. If for some reason it's predicting a big infection period and you don't even see rain or a cloud in the sky anywhere and it looks like it's going to miss you on the weather radar, maybe don't do it this time. And always try avoid spraying after an infection period. This is where things get resistance. You could let this all infect and then spray there on 529, but the smarter thing to do is to see that there's going to be a week of rain coming and know that it's probably going to predict some pretty wild Ascaspore infections, and really get it there and then come back in if you need to later with a light. You could probably even get away with a biopesticide if you used a really good single site fungicide in this particular instance, and you'd probably never even see apple scab. But spraying here is nice because you can get a little protection for that. You can go back for that, but that puts a little more stress on the resistance selection. With that, we'll finish up apple scab.